Welcome everyone to the plenary lecture to be given by Professor Susan Holmes. My name is Miguel de Carvalho. I'm a reader in statistics at the University of Edinburgh, and I'm also the president of the Portuguese Statistical Society. I'll be sharing this session. I'd like to get started by introducing our speaker, Professor Susan Holmes. Susan is a prominent statistician who is a professor at statistics at Stanford University, as well as a member of BioX. Professor Holmes' research lies at the interface between mathematical statistics, probability, geometry, and biology. Susan is a world authority on statistical and mathematical modeling of complex biological phenomena by applying and developing ideas, concepts, and methods that stem from geometrical statistics, non-parametric multivariate analysis, bootstrapping, as well as data visualization. Among her many distinctions, I would like to highlight that Professor Holmes is a fellow of the Fields Institute, as well as a fellow of the Institute for Mathematical Statistics. In today's talk, Susan will be speaking about statistics and geometry for biology systems. Welcome, Susan.
Thank you so much for inviting me. And I think it's wonderful to have this interface, I hope, and where statisticians try to talk to some mathematicians. And I'm just going to show some of the examples of how mathematics come into my work. So to begin with, I would say that what I try to do is I try to bridge statistics, biology, and mathematics. And this is a bridge in Portugal. And I think the Portuguese are the champions of bridges, so they understand the difficulties. When you build a bridge, you have to build from both sides and you have to meet up somewhere. And that's one of the big difficulties in some of the work I do, both from the biology side and from the mathematics side. The particular areas in which I work are the microbiome, which I'll talk to a little about, and immunology. Both of those involve very complex systems with very high dimensions and heterogeneous data. And so in that, we use dimension reduction methods. Some of them are linear, some of them are nonlinear. We use densities, probability densities, and things like duality. The standard duality which occurs when you use inner products. Heterogeneous data integration is a main part of modern data where we have the integration of multimodal data. We often have trees, phylogenetic trees or networks between observations, which require a change in the metrics. What we're after when we try to analyze the data are the discovery of latent gradients or orderings. And I'll try to show how this can be done using geometrical methods. And then at the end, as statisticians, we try to do inference. And we have to be honest about how uncertain our results are. So uncertainty quantification is best done in a framework in which we do Bayesian posterior distributions. So those are probability distributions, which we can compute over complex spaces. And I'll talk a little bit about the challenges which the results that we are provided by these methods pose. So the data that I'm going to analyze has come from joint work with large grants that I have with David Relman and his lab. And we studied the resilience in particular of the human microbiome after perturbation. So when you take antibiotics, or when you're sick, or when you have a colonoscopy, or when you change your diet, that's a perturbation which actually has an effect on how the microbes in your gut are reacting. And so I'm going to talk a little bit more about the data. And to start with, many multivariate methods start off as just simple rectangular matrices. Matrices are actually not enough for our types of data. So I would say there's a system failure if you just suppose that you have a matrix of data. We have to go further than that. And to give you an idea a little bit more about the structure, here is a presentation from a paper which I wrote with my group on the representation of longitudinal data as it's measured after people are taking antibiotics. And then we have the phylogenetic tree, which is represented in a radial representation here. And we have the abundances of the different bacteria. And you can see that particular parts of the tree have high abundances. And then you can see how much they change in these what we call spark lines. So as a statistician, and if any of you have studied standard probability, you know that the basic modality is to suppose that we have samples which are IID. And we don't have samples which are IID. They're neither identically distributed nor independent. The other difficulty is that, as I said, the matrix setup is actually not enough because a matrix has a finite number of columns. And in our case, features are not predefined. So the best way of thinking of this mathematically is you actually have infinite number of columns. At a micro level, in some sense, data are not homoscedastically distributed. That means that they're not distributed so that they have the same variance across a whole manifold. So we very often like to think that there's an underlying manifold which explains a latent biological phenomena. And that might be a fitness landscape or an ecological landscape in which certain things are optimizes. But what happens is, if you had uniformly distributed data on the manifold, it would be actually quite easy to use some kind of smoothing to obtain the manifold. But we can't do that. We can't do that without doing various kinds of transformations. And so homoscedasticity means that you have the same variance across all the areas where you collect the data. And this is not true for biological data. So let me give a little bit more details about the actual data that we collect. We have DNA signature barcodes. And this is small reads of a length about 300 base pairs long, which are like fingerprints of certain species that we can collect. So we know the abundance, or at least we have an idea of how many reads correspond to these different species or taxa. Another thing that we also measure through reads is genes and how they're expressed. And so we have what we call RNA-seq gene expression data. We can measure what metabolites, what chemical components are present in the sample. And this is measured with what we call MS spectra, which are represented here. We also have data which are a priori data. That is, we know about the phylogenetic tree of the different, say, strains of bacteria which are present. We also know, we have prior knowledge about the metabolic networks which involve the metabolites which are involved. So we can't ignore that prior information, and it's really important to incorporate it. But that's one of the challenges of what we do. So how can we involve all the prior data in the analysis of the counts, the reads? So we use graphs or trees to influence distances that we use for our low dimensional representation. I would say in some sense that we have structured high dimensionality. We have to take into account this difference across the manifold of the distributions, and we do that using mixtures. So we have mixtures of distributions. We don't just have a normal distribution. We have many, many distributions which are all mixed together. It can be very instructive in terms of interpretation to end up with latent variables or factors which explain the phenomena we're going to see. And I'll show a few examples of this. As a computational statistician, we don't have to stress about the choices we make, because you can come back if you use a reproducible workflow, and you can change your choices and make a different choice of distance, or incorporate different data, or drop some of the outliers. And so this is enabled by having a reproducible workflow. So in statistics, it's very important to separate out two things. One is the data themselves, and the other is the model, the ideal model for the data. Now we go from the data to an estimate for the parameter. So the ideal parameter would be the Greek letter theta, and the data gives us the estimate, which is the theta hat. And from that estimate, we can go to a probability distribution and our best guess of the unknown parameter, which parametrizes. Now, as I said earlier, the number of features is often not finite, so this theta can be infinite dimensional, which is what happens in non-parametric statistics. And I'm not going to go into too much details. There's a book online that I wrote with Wolfgang Huber, which goes into all the details about this paradigm, which I think is important, because people very often confuse the data with the parameters themselves, and especially from a geometric viewpoint. That is, sometimes the parameters theta belong to the simplex, but that doesn't mean that the data do. So what are the data? Well, just to give you a more concrete idea, we have different samples. So they might come from different subjects, or they might come from the same subject measured across different time points. And then we have different strains. And these strains say we had 81 reads that came from the sample 1, and 22 from sample 2. If we look at the sums of the column, so in sample 1, we'll have a very different number of reads than in the column sample 2. So that difficulty is what brings about the heteroscedasticity. That is, we don't know the estimates with equal variance. The number of ASVs themselves is not fixed a priori, so they can be infinite. The number of samples is finite. So as a first approximation, we say, okay, suppose that we are going to restrict ourselves to a finite set or a finite number of taxa, and what we're interested in, for instance, in a control setting, you have a proportion of the different variants or species. That would be one vector. And then I is the number of these strains. And then you have another vector for the treatment. So we're interested in the difference between these, and we're going to need to make inferences about this. So we need the uncertainty. And this is one of the big difficulties that we have, is the difference in sample depths, and we have to deal with that as an uncertainty. Now, I'm going to make an aside about how we deal with these problems by coming back to geometry. So we have different levels of geometry that we do for the data. And to start with, I'll start with a Euclidean setting and explain what I mean by the geometry of data. So each one, if you take the observations, you can suppose that each one of the points here corresponds to one sample, say, and they're measured in feature space. And so the features are the vectors, the underlying vectors. There are many of them. And here you have, I'd say, a cloud of points. So the geometry of data actually occurs in two spaces. One is, as I said before, you have the points which occur here. And here I've actually assigned them different sizes because they could have different weights. But you could also say, okay, I can look at the jewel and say, I'm looking at the features as they occur in the coordinate space of data points. And that's just a fact that we have duality. We can look at the dimensions in two different ways. And we have one inner product, which corresponds to the inner product between points as they occur with weightings on the variables, and the other one with weighting on the observations. In fact, this duality is something that statisticians are well aware of, and sometimes we talk about what we call transposable data. That is, if your samples weren't drawn IID from a certain population, there's no reason to give priority to one framework in terms of your duality as opposed to the other one. And as I said earlier, the number of features is actually so large that you could say, I had an infinite number of features and I drew the features at random. And so this duality is a useful way. I'd say that the data are variables measured on n observations, and we have n rows and p columns, and the columns are the features. And we have a matrix of weights on the observations. It might have to do with some prior knowledge about how they're connected or how trustworthy the results are. So sometimes you might have some of the data come from a lab which is very trustworthy, and so you want to put a high weight on those, and you might want to downweight some of the observations which you're not so sure of. You have weights that you put on the variables, which have to do with the fact that all the variables don't have the same variance, and so you could make them so that they do by just putting in a rescaling. And I think that as we go towards using distances, I want to remind you this very important point, which is the reason that some of the geometry here is a bit complicated. And that's the fact that distances aren't everything. You have to incorporate the baseline probabilities. And there's this wonderful book by Danny Kahneman about thinking fast and slow, which explains that we forget about the baseline. And this has been a big problem in biology, where many sort of simple bioinformatics methods actually abstracted and say everything is uniform, and they forget about the baseline, and then you get the wrong answers. So that's why in our weighting, we do want to put in probability weights. And so you could do it at the level of the inner product, which I show here, which gives you this duality. And we'll see later that if you don't have a Euclidean space, differential geometry allows you to do that better. So first of all, in the linear world, you can do it just by doing a generalized principal component analysis, where you incorporate the weights in the inner products. And so in a standard singular value decomposition, you obtain USV prime, and you have this the orthogonality condition, which incorporates the weights, both on the point n dimensional and the p dimensional spaces. And this is one of the ways that we can put in our geometry some of the probability. And as mathematicians, you might appreciate my advisor in France, Yves Escoffier, was a big proponent of doing this using algebra and using commutative diagrams, and saying, okay, we have a first commutative diagram given by the data, but then what you want to do is find here the dimension is p, n by p. And so the dimension of this matrix V, for instance, is p by p. And you want to find a best approximation, but of a lower rank. And you can say, okay, I'm going to take this commutative diagram and give an approximation of it with a lower rank. And that's obtained by doing the eigen decomposition. And so you get a rank of the diagram, and the theoretical rank, of course, is lambda r. And then you can get an approximate one by just taking the first k largest eigenvalues, and you get the best method to approximate that in general. And these have been extended, this approach has been extended in kernels and machine learning nowadays to more general phenomena. So when I say we approximate one diagram by the other, there's actually a concrete way in which we measure the quality of the approximation, and that's through what's called an RV coefficient, and it's just an inner product between the operators which correspond to the two diagrams. So it's a generalization of the ordinary inner product between vectors to the inner product between the operators associated to the diagram. So it's in some sense the cosine of the angle between matrices, and we define the approximation in terms of this RV coefficient, and this Frobenius inner products allows us to compare and find out what the best operator is. Now, this inner product is actually useful at many levels, because as I said earlier on, we often have multimodal data. And when you're doing that, you actually want to look at the inner product between matrices, not the inner product between vectors. So this inner product will come back several times. But it particularizes if you have two matrices which are just vectors, it becomes the square of the ordinary correlation coefficient. Now, how do we use this? I said at the beginning that one of the really important things about statistics is that we don't only give low-dimensional approximations to data, we actually provide uncertainty quantification. So here's an example from a paper with Lan Huang, where we were trying to show how you can take ordination plots and bootstrap the data. That is, you resample the data many times, say 1,000 times or 100 times, and then give an approximation of the uncertainty using the bootstrap. Now, there are methods which require that the data be multivariate normal, but in this case, we wanted to use a non-parametric method, which doesn't suppose that the data are normal. And so in these two examples, we take two-dimensional representations of, first of all, data which were originally of low rank. And you can see this because 100% of the variability of the data is actually contained in the first two components. And here, this is data which has a higher dimension. So the approximation is weaker in some sense. There's more uncertainty about the approximation. Now, we had a real difficulty when we're trying to bootstrap this data, and that was that, as you know, when you look at the eigenvalues and eigenvectors, the eigenvector, first of all, the definition of the coefficients of an eigenvector are not well identified. That is, you could have a plus sign or a minus sign in the coefficients. But it's worse than that because often you have inversion. That is, one eigenvector, if you have eigenvalues which are quite close, the first eigenvector might become the second one. And so you have this switching of eigenvectors. And so when you're doing a bootstrap, it actually makes a complete mess. So here is just a small example where we did only 5 points and we resampled, and you can see that then not well aligned. That is, they don't project together. And the reason they don't project together is what I was saying. That is, here you have this point, some sample one, sometimes it's positive and sometimes it's negative. And then sometimes it's associated to eigenvalue 1 and sometimes it's associated to eigenvalue 2. And so the coefficients are not well identified. So to overcome this, we actually use this RV coefficient I was talking about. We compute the coefficient, the RV coefficient between the different bootstrap resamples of the operators that correspond to the commutative diagram. And then we take that matrix and we find the first eigentable in some sense of that. And that allows us to align all the different tables. So what have we seen up to now? Well, we've seen that in a Euclidean setting, we can use the duality in the data we can do a generalized singular value decomposition of the rows, or a generalized eigenvalue decomposition of the columns, depending on which one is actually smaller. So the duality is beneficial. If you have a really large number of features, you're better off doing the eigenvectors of the Gram matrix WD. We've also seen how this angle, this RV coefficient between operators, allows us to create a consensus to compare projections. Now I'm going to do a little bit more about how then we can change some of the metrics to use prior information. So what's the motivation? Well, here is a few of the phylogenetic trees of the different bacteria involved in the microbiome at different body sites. And so, for instance, in the oral microbiome in the mouth, we have a certain number of bacteria. In the intestines, we have different bacteria. And we can make the tree of these bacteria, and we plotted here the pink dots for the species that were present in this person. And you can be interested in representing the data, the abundances, with taking into account the similarity on the tree. So we want to incorporate the trees with the counts. So this is work that I've done with two of my PhD students, Elizabeth Purdom and Julia Fukuyama. And the idea is, we have prior information on the phylogenetic tree, which is a rooted, binary, semi-labeled tree. Here you have the strains, and then we have samples, and we want to represent the samples using the tree information. But the first thing we do is we take the tree, and we can compute a distance along the tree between the different samples, between the different strains. We use standard multidimensional scaling, which is a method from going from a distance, even if it's not a Euclidean distance, to an approximate ordination or embedding in a Euclidean space. And then we use these points, and we take the barycenters or the centroids with regards to a weighted masses, which have to do with how many reads of each of these different species were available. So let me go through some of that a little bit. First of all, I'd say the main workhorse in the world of ecology or multivariate statistics is the use of Euclidean embeddings. And so any distance, it doesn't have to be a Euclidean distance, we can make a coordinate space which has a certain number of Euclidean coordinates where the distances between the points in this Euclidean embedding are quite close, or as close as possible to the distances that you're given. So you could start off by a distance between trees, which is a distance in a cat-0 space, and you can still make a Euclidean approximation to those distances. And of course, statistics has the benefit that we're not like mathematicians. We don't have to get everything completely right because we have very noisy data. And so if you get a Euclidean embedding, which is of better quality or has lower approximation than the amount of noise you have, then it's just as good. So we can go backwards from a distance, so I'll call this distance delta, to the Euclidean embedding, doing an eigendecomposition of a doubly recentered version of the square of the matrix. So the square in this case is we take the square of all the different elements in the matrix, and we have a centering matrix, which is the identity minus 1 over n 1 prime. And we apply it on both sides, and this gives you, when you decompose it, the best rank R approximation to the data. Now, this was actually already known by Schroeder, who asked the question, how do I know whether this distance is actually a distance between Euclidean points? And he actually used this matrix B for doing that. And later on, Eckhart and Young said you can go further, you can find the best approximation. So this is done with, again, singular decomposition. And all the algorithms that we use when we're doing multidimensional scaling use this algorithm. So we usually use the number of dimensions by looking at the eigenvalues and deciding when there's a cutoff in the scree plot. And of course, as I said, the advantage is that the original distances aren't Euclidean. So we're going to use this method twice, actually. We use it first with the distances along the tree that I showed you between the species. We find an embedding, and then we use that embedding, but we weight the species according to how much they occurred in the samples. And then we do another ordination, and we take the barycenters and we provide multidimensional scaling of this. Just a word on vocabulary. This is called double principal coordinate analysis, just because the embedding method has two names. It's called multidimensional scaling by the statisticians, and it's called principal coordinate analysis by the biologists. So double principal coordinate analysis is the first name that it was given to do this. And I'm just going to show you an example. Here we have the samples in our study, were different samples of the gut microbiome. On patients, there were only 3 patients in this preliminary study where we gave them 2 courses of antibiotics, and we want to see what the differences were in patients under what we call antibiotic stress. So we categorized the times as before taking antibiotics, during the antibiotic period. There's a week between courses of antibiotics, and then we have the second antibiotic. So the first antibiotic in the first week after, and the second antibiotic time and the second week after, we call the patients being under antibiotic stress. And what we represent here is the different samples. So the first thing we do is take the species and do the multidimensional scaling, and this is what you get. And of course, your representation, the data originally sit on a tree, so the representation is very tree-like, as it should be. But then we take the centers of gravity, we do the projection, and here we see that we have 3 colors, and they're the 3 different patients. So in some sense, the first effect that you could see is there's a within subject effect. So subjects vary, and the blue subject here has much less variation than the green and the red, and we'll talk a little bit more about that in a minute. And then here, this is the representation of the different bacteria and which ones corresponds to being more to the right or more to the left. Now, what we're actually interested in is the antibiotic stress. So we're going to just make 2 groups of samples, those that were stressed and those that were not stressed. And we're going to look at the difference. So we see this subject F, who I pointed out before, they're very resilient. They don't have hardly any change when they take antibiotics. Somebody who's not affected by antibiotics. And the 2 other patients, there's quite a big gap, mostly towards the left. So the perturbed ones are the D2 and E2. And so we have an understanding of what's going on in this very high dimensional data just by looking at this plot, this representation. So this is a case where this projection was very useful. Now I'm going to switch a little bit up, because although I use a lot of ordination methods, nonlinearity is actually the norm in the data that we see. And this is a data set here that I'm representing, which is the gut microbiome of many, many samples across the world, in which we took different peoples from different countries. And so there we have some native peoples from places which are not at all touched by civilization and have a very traditional way of eating. And then here we have, towards the end, we have Italy, Australia, USA, Canada. And you can see that those are the ones that are all on the right here. And they have a very different profile of their gut microbiome. And what I'm interested in is actually this gradient. So what you have is, of course, you could say that it's a multidimensional or tridimensional manifold to start with. I'm going to just do the simplest case where I say that there's an underlying manifold here, which is just a gradient, so it's a one-dimensional manifold. And we're interested in this one-dimensional manifold, which has a real meaning. It's a latent variable, which appeared from the data and which we could interpret as a departure from traditional food. And this is something which has occurred multiple times in microbiome data. We first saw it when people started doing the microbiome. They were thinking that maybe the best way to analyze the data was to find clusters. And that means you find a latent variable, an underlying variable, which is categorical, which takes only 3 modalities. But in fact, the underlying variables here are continuous. It's a continuum. And so we have data on manifolds, and generating data on manifold is not simple. That is, we have a paper with Fatih Ayakonis and Mehrdad Shashahani that shows that if you take a uniform distribution over the underlying parameters, you don't get a uniform distribution on the manifold. So it's very important to take into account the local volume. And when you're generating on a manifold, that local volume allows you to get the true uniform distribution. But I'm going to be interested in the inverse. That is, I know the data aren't uniform, and I want to go back to something which is closer to uniform. So here's what we did. Here's an underlying gradient, which is very easy to understand. These are all microbiome studies. These are samples of bacteria. They were collected, metagenomic samples as they were collected in the ocean, and at different depths. So here are the samples. These are all the points here, the gray. And then we found that the first underlying structure, if you make a representation using any kind of ordination or local ordination method, you get nonlinear representation here along this curve. And if you color along the curve, you find that actually the underlying phenomena is the depth at which the samples were taken. And of course, the biologists, when they see this, say, of course, the depth is the first thing because different bacteria thrive without light and in different temperatures. So at the surface, the water is warmer, there's much more light, you have a certain class of bacteria, and here you have another class of bacteria. But as I said, one of my goals is not only to give a representation of the data, it's also to give uncertainty quantification. So here we wanted to project on the gradient, the underlying gradient that we perceive here. So these points are projected down, but giving us an ordering. So the result is what we think the depth ordering is with uncertainty. So we're doing this projection. And this is a little bit what we're trying to do. So as I said before, the data are not uniformly distributed on our manifold. So here, for instance, you have great big gaps in some of the areas, and that's a difficulty. The clustering can trick you. And so what we're going to do is we're going to rescale the data locally in the same way that you would do in a Riemannian setting. That is, that you have local maps, and we're going to try and make the local maps stick together. So this was done with Lan Wang-Nguyen. And the idea is to use a prior distribution And we call it Bayesian unidimensional scaling. It's not multidimensional scaling because we're really scaling along a trajectory. And so this is the 3D representation of that same trajectory that I appeared. And so we start off with pairwise dissimilarities, and then we're going to go to this one-dimensional curve. So we suppose that the original true data are on this manifold. So on a latent manifold with a parameter, which is just one-dimensional belonging to 0, 1, which I'll call tau. And then there's a true underlying distance with a delta ij, which is the distance along the manifold. And we put prior distributions. If we know the true delta ij, then we have a distribution uncertainty, which occurs in the measurement of the distance and the biological variance. And we put priors on tau and on the variability, so quite a wide distribution. And I won't go into the details, but this is the way that as a statistician, we set things up with a prior distribution and we get a posterior. So we're going to use the distance heteroscedasticity to try to change the measure so that we can bring things to being not perfectly homoscedastic or perfectly uniform, but closer to that. And we use the data to do that. So that is that we take the points and we make an estimate of the local variances using distances between the points that we have. And so this is how it works. We use Stan, and this is how we built the confidence contours. That is, we get a posterior distribution with our model, and we need to put it all together. And we know how to do the uncertainty contours because I already told you about our V coefficient. And so we can put that together and compute all the different distances, and then do this R0, this RV coefficient, and project everything down on the consensus. So just to say about what we did for the manifolds, we can use the points to estimate local variances, and it's equivalent in some sense to seeing the local variability and the local curvature. We rescale and reweight using the distances, and we combine the posterior picks from the samples, and we use our consensus registration method that I talked to you about before. So that's the one step forward. That's the how to deal with nonlinearity. And then I would just like to say a little bit more about how you can do what we call a full Bayesian non-parametric model. So when we don't know the total numbers of features. So I said that actually the truth is that you have to suppose that your features come from an infinite set of possible features. And that's because in statistics, infinity starts when you have more features than you have samples. So if you only have 100 samples, as soon as you get more than 100 features, then you have infinity. And so what we suppose is, as in the case of the depth variable, we have, in fact, underlying low dimensional latent variables. So we're supposing that we have some low dimensional manifold that we're trying to discover. And we also use the fact that the bacteria are not independent. So some of the bacteria can't live without other bacteria. And so we can't suppose that they're independent. And that's actually the explanation of the low dimensionality. But we can use that as our marginal priors and give discrete distributions. So this is a diagram of what we're trying to do. In this case, these are different species. I call them the OTUs. Here I have I of them. And then I have these different samples, or the populations in the samples, which are assembly of communities. And these populations, there are J of them. But I'm going to set things up exactly as I did before. That is, in the case of the ocean data when we were doing depth. And I'm going to generate many, many samples according to a Bayesian posterior. The parameters that are given are they're unknown, but they're part of the model. And we suppose that there's some amount of noise. So we have an inner product between the underlying key characteristics of the samples, and then we have some noise. So this method and this way of thinking was published in the Journal of the American Statistical Association in 2017, and I did this work with Sergio Baccalado, who was a postdoc at the time, and Lorenzo Trippa and Bo Yu Ren. So the low-dimensional factors, so if we look at, say, a small toy example, the way we see it is we have the species, and then the abundances of the species gives you a point according to the proportion of each of the species, but also according to their association. So some of the species are very correlated. So the vectors in the multidimensional space have a very small angle. And so in this case, we have the vector for these populations are quite similar. So you see them here, the abundances, the vectors themselves have a small angle. So most of the time when you see the yellow one, you see the blue one. And on the other hand, you have orthogonal ones, which don't occur or don't co-occur. And so you see as if some bacteria exclude other bacteria. And this is one of the reasons why in the real world, we can't use the multinomial as is for these types of methods, because in the multinomial, apart from the number of reads that you have, the balls in the boxes would be independent, which is not what occurs. So the parameters are these factors, and they're supposed to be these underlying low-dimensional factors that we have. And the degree of similarity between the distribution is measured through this angle that I showed you a picture of, these phi j, j prime. And then we have we build up a dependent Dirichlet process so that we have realistic distributions, and we approximate it, the arrays, so that we have, we build it from Poisson processes, in fact, with different intensities. And I won't go into too many of the details, but what happens is that we then, what we're doing here is building up every single new pick from a posterior distribution, which is inspired by the data, but we're going to act as if we could generate, we have a generative model, and we could generate many, many instances of this posterior distribution, say, as if we could generate many different data sets, and then we have to put them back together. And as I said before, if you try to put them back together and don't take into account the identification of the different directions, you get a mess. So this is the first plot that we made when we created a posterior distribution for a given data set. And we had actually, 5 different types. And we were trying to get projections of those 5 types. And then again, this is exactly the same problem I talked about before. We have to do registration. That is, you have to register the data so that you have a consensus space, a compromise, where everything is the same relative. So again, we use this RV coefficient. We compute cross correlations between the different tables that we got from the posterior distribution. Here we're using what's common to all of them is the OTU sets. And then we have these different cross correlation matrices, and we compute the RV coefficient. Then we compute the first eigenvector. And it's the first eigenvector which serves as the consensus. And when you project onto that first eigenvector, and again, the table for which we compute the eigenvectors is the one with the RV matrix, this is what you get. So we went from that mess that I showed you before to this representation where you can very clearly see that most of the points, the uncertainty is very, very small. So we have an uncertainty in our representation, in our projection, which is very good. And we are quite sure about the projections, except for these 2 points, which are very uncertain. So we went back to these 2 samples and tried to find out what was specific about them. And it turns out that these 2 samples had a very low depth of the library size. That is the number of reads. And so that was the sum of the column, and was much smaller in the others. So they're known with very high uncertainty. So the variability, the variance. And so this is exactly what we want, because we want to be able to program or design another experiment, a follow-up experiment, where we say, these ones need to be reanalyzed. These ones we're all sure about. So this provides the uncertainty quantification. Okay, this was another data set in which we're trying to actually look at types of IBS. We can see in this case, the first 4 were of one type, the other up to 16, the others were all of the up to 22 were all of the same type. And we see actually that 16, which was supposed to be of the same type as this, is much more of an intermediary case. And so there's a lot of information in looking at the uncertainty contours. Okay, I want to finish off by saying that what we learn when we do this kind of thing is you have to incorporate all the information that you have. And I'm very inspired by Don Knuth, who is, of course, a computer scientist, but also a mathematician. And he has this line, which is very important to me because I do a lot of coding, which says that premature optimization is the root of all evil in coding. And what I'd say is that premature summarization is the root of all evil in statistics. And what I mean by that is that we have a tendency to say, okay, I'm going to boil everything down to just distances. And I'll forget about the baseline probability measures underlying, and I'll be pretending that I'm dealing with a uniform distribution on a manifold or on a space. So people do that because it's very hard mathematically to take into account distances and weights at the same time. We'd like to ignore the fact that we had this tree relationship between species, or ignore some of the information. So we're trying to summarize too early. And I think that mathematics is the main tool that can avoid this premature summarization. That is, the simplifications that we do are due to a lack of tools. And I would definitely, encourage mathematicians to talk to statisticians, because differential geometry is definitely the way forward for much of what we do. And although the formalization of differential geometry has been very nicely done in what's called geometrical statistics by people like Xavier Penick and other collaborators that with whom I work, who work on shape spaces. But in standard statistics and data science, I don't think there's a lot of use of differential geometry, and it's a shame. So what I learned, as I said, that distances and probabilistic weights are keys, but you have to find ways of melding them. We need different approaches. That is, on one hand, linear approximations can be enough, but much of the data is nonlinear, so you do need manifolds. We don't only use one distance. We have at our disposal hundreds of different distances, which incorporate different types of information. And there are many ways of approximating the data or rounding it. And we have to own up to choosing at one point, I'm doing a linear approximation. Here, I'm not doing a linear approximation. I'm doing a Euclidean approximation. I'm not using a Euclidean approximation. I don't think we're clear enough about how the approximations are being used. I like to use reproducible research, so this liberates me because my choices are not forever. If you save the sufficient statistics in the code, you can go back and change things. And then I have many questions for mathematicians, and I'd be encouraging of you to ask me questions about the statistical part. And so I want to thank my lab group, the people I've worked with. Here they are. Many of the biologists are very patient with me because usually in biology, you have a question, you're supposed to answer it within a week or so, and sometimes it takes me years to answer the questions. And so they're very patient. I've benefited from tools because all the computational work, which I haven't shown you, rests on the use of reproducible research, and in particular for the Bayesian work, it's STAN, the rest is R, and then a bioconductor community. And then I'd like to thank the Portuguese Math Society for the opportunity to talk to you about applications, but I also want to thank the Portuguese people for their very warm welcome over the past year. Okay.
Thank you, Susan, for this amazing talk. The floor is now open for questions, comments, and remarks. Please. Just unmute yourself and post questions. If there's no questions, I'll break the ice first with the first question. So it's a general question. You were mentioning RV quite often, and so RV is appropriate then for matrices. Matrices are natural for approaching images, but there's also much of your work which is developed also over other types of data, such as trees, networks, etc. Shapes as well. So how do you see all this world based on this geometry of RV also extending beyond that?
So the RV is the simplest to explain because, again, it's linear and I use eigenvectors. But in fact, that's where the differential geometry comes in. That is the problem of registration and the work of Xavier Pennec and many people in the interface between differential geometry and statistics is this work of, you think about, so for instance, working on brain images, you have many different brains of different people, and you want to make a consensus. And the consensus between the different brains has to take into account that the shape of the brain, there are areas very high variability. And so the differential geometry allows you to make atlases. So this is definitely where statistics comes in. The interface between differential geometry and statistics. And so I presented something very simple, but people have more general coefficients, generalized, which are not linear. So because you want things which are local, but there's still a huge area at the interface of mathematics and statistics, which is open.
Okay, so feel free to add your comments, questions, or remarks to the chat.
So I don't see the chat, so you should.
I'm seeing the chat.
Okay, that's good.
I'm seeing the chat, and just providing an extra incentive if someone has more questions, actually, to ask them. So here's another question then. So in terms of modeling manifolds, at least conceptually, for some situations, perhaps it makes sense to model pairs of manifolds. Has there been much work in terms of modeling manifolds from a joint perspective, building d-dimensional distributions over manifolds?
Well, I think that so usually when we have two manifolds, the most common thing is to try and make mappings that make them correspond one to the other. And so the mappings, the type of mappings that we try to make, and maybe I have a short picture of that here. So it might be, it's not necessary. I mean, I think that the hardest problems is you have the two spaces here, the two mappings, which are separate. But then you need a common one, which you go back to, and then you have to have an atlas here where you have one where there's an intersection. And the idea being that if in these two spaces the distributions are different, you can go back to a manifold where you've changed the local geometry so that you've spread things out, so they're much closer to being uniform. So I think that the two manifold, or you have these two sets or two comparisons, the mapping is hard in the here because you have local densities which are very, very different, and then you have to map them back to a manifold where they have a overlap. And so this all comes from using standard differential geometry, but I think that that's starting to be very productive, but it's an area of research which is wide open. So there are really very, very nice problems in mathematics, which have to do with the fact of how can I make it so that the data are at least closer to uniform on the higher manifold than they were on the lower one, on the data. And again, as statisticians, we're allowed to do approximations. It doesn't have to be perfect. The embeddings that we choose, as long as they're better. And that so we have less constraints than if we were just doing it mathematically.
Okay, so just a final question as we're running out of time. So the standard trick in a univariate situation to move data into a uniform scale would be to actually compute the distribution function evaluated at x.
Right.
Something like this would still apply on this space?
Yes, so that's the idea. That is exactly right. That is the functions that we use are approximations. Now, technically, there are impossibility theorems that say you can't do it exactly in most cases for statistics, but we don't want to do it exactly. We want to do it at least better than the original, so more uniform, but it doesn't work exactly. But that's exactly the idea of these inverse maps, right?
Okay, thank you. So let's thank Susan once more for her excellent talk. Thanks also to the organizers, and that closes today's session. So thank you, everyone.
Thank you very much. Thank you very much.